Based on WHO's 2014 data, 1.9 billion adults were overweight and 600 million adults were recorded clinically obese. And based on the records of WHO 2013, 42 million children with ages 5 and below were recorded overweight or obese. The central nervous system plays a vital role in maintaining the balance in our body we know as homeostasis. And within the brain, the hypothalamus has an important role in integrating and regulating homeostasis, including energy metabolism regulation. Within the hypothalamus, one of the key hypothalamic nuclei that regulates the energy expenditure is the arcuate nucleus. The hypothalamus contains two types of neurons: orexigenic, the appetite-stimulating, and the anorexigenic, the appetite-suppressing. The appetite-suppressing neurons project to the second-order neurons that manages the energy intake of the organism. Damage on the paraventricular hypothalamic nucleus leads to overeating and obesity. Damage on the ventromedial hypothalamus leads to hyperphagia and obesity. Damage on the dorsal medial hypothalamus and lateral hypothalamus produces a hypophagic and lean phenotype. The extrahypothalamic neuronal circuits are connected to the mesolimbic reward system and the regulation of energy expenditure, where other brain regions like the amygdala and striatum assist in the production of dopamine to stimulate the reward. Once a person has decreased amount of glucose in the body, like that of during fasting, glutamate and appetite-stimulating neurons are then activated, which stimulates dopaminergic neurons. Similar to this path is the activation of inhibitor GABAergic neurons that increases the food intake and consumatory behavior, where there is a drive to receive a caloric reward from a compulsive sucrose-seeking behavior. With regulating the glucose energy levels, two main hormones are used: leptin and insulin. Insulin controls the glucose metabolism by suppressing the hepatic glucose production, and an important factor in maintaining glucose homeostasis in the hypothalamic level. Leptin derived from adipose tissues were induced by insulin and plays an important role in the regulation of food intake, as it inhibits the food intake and to increase energy expenditure, which coincides with the appetite-suppressing neurons. Another important regulator of the central nervous system dependent energy control are the gastrointestinal hormones. Ghrelin is secreted in the stomach during starvation and stimulates feeding by activating appetite-stimulating neurons, and in addition, activates the paraventricular hypothalamic nucleus, which promotes body weight gain and adiposity. Other hormones exert appetite-suppressing effects in various brain regions and by modulating vagal afferents. Hypothalamic inflammation and insulin and leptin resistance is often associated with obesity, as the ingestion of high-fat and calorie-dense diet with increased amounts of saturated fatty acids would cause inflammation of the hypothalamic neurons. This inflammation impairs the effect of insulin and leptin, contributing to hyperphagia and development of obesity, along with the dysregulation of glucose homeostasis. Several studies now focus on the effects of obese women and their association with metabolic disorders of the offspring. In rats, a maternal diet, which is rich in saturated fatty acids during gestation and lactation, leads to hypothalamic inflammation in the rodent offspring and non-human primates, and the projections of neurons were altered, resulting in the increased obesity and impaired glucose homeostasis in the offspring. The mitochondria is well known in regulating metabolism within a cell, and now it is seen as an important factor in different metabolic disorders such as type 2 diabetes mellitus, obesity, and cardiovascular diseases. Mitofusins are GTPases that are important in regulating the processes in the mitochondria. Mammals have two mitofusins, MFN1 and MFN2, which are seen in rodents to protect them from high-fat diet-induced obesity. Deletion of MFN2 resulted in leptin resistance, hyperphagia, reduced energy expenditure, and morbid obesity, while the deletion of MFN1 did not alter the metabolic state. Gene expression can also be a factor in hypothalamic disorders, as mutations in certain genes of receptors, like MC4R and POMC, could lead to hyperphagia and obesity in humans. And it was also observed in humans and mice that deficiencies in the gene coding for leptin or leptin receptors resulted to severe obesity. New research now covers the effect of uridine diphosphate or uridine levels circulating in different neuropathways that promote cycles of weight gain and exaggerated food intake. With a clearer view on how the central nervous system affects obesity with the dysregulation of energy and glucose homeostasis, therapeutic strategies like using polyagonists and other metabolic hormones to exhibit higher efficacy in dealing with the body weight reduction and glycemic control with less side effects compared with similar amounts of monoagonist doses in rodents and in humans. The use of the MC4R agonist has been seen successful in the treatment of POMC-deficient obesity, as well as an effective treatment for humans and non-human primates with non-genetic obesity. Obesity is a growing epidemic and is correlated to the metabolic disorders that are controlled by the molecular mechanisms in the central nervous system. The study of the hypothalamus and important key factors like the hypothalamic nuclei and the receptor pathways that work inside helps researchers understand the feeding control of an organism and the reward systems that can be integrated in developing specific therapeutic targeting that help in the fight to decrease obesity cases in the future.